is Professor Brent Mitcheller. He is the president of CNPS's newest chapter, the Bryophyte chapter, which is the only statewide chapter. Every other chapter, to my knowledge, is geographical, such as Marin. But Bryophyte kind of paved a whole new concept for CNPS. I was on chapter council when this was debated, and it took some doing, and we've done it, and I think everybody is very happy with it. Professor Mishler is the distinguished professor of the Department of Integrative Biology. He also wears other hats. He's the director of the University in Jepson Herbaria at UC Berkeley. And it's with great pleasure that I turn the meeting over to Professor Mishler to talk about mosses are from Mars, vascular plants are from Venus.
Great. Thank you very much, David. And thanks for inviting me, everybody. I do love CNPS, and I always have, and I really appreciate what the chapters do. So I'm always glad to speak to other chapters. All of our Bryophyte chapter members are also members of their local chapter. So I'm active in East Bay chapter. And I think the chapters are what makes CNPS really special. So what I'm going to do after that, thanks, is share my screen. And once you guys can see my screen, which I think you ought to be able to now.
Yes.
There we go. Okay. Well, so what I'm going to talk about, I do have many interests, but my center interest and my favorite group of organisms are Bryophytes, even though I do like all plants. And I have nothing against vascular plants. But the main point of this, and it's funny that David called it Moses to begin with, because my master's thesis, the title of it was Mosses of the Chaparral, but it's printed in the program and in my diploma as Moses of the Chaparral. The university even got it wrong. So we're talking about mosses today. And what I want to convince you of is that they're really different than vascular plants and really different than the kind of biology you think of normally when you think of the larger, more garish flowering plants. So what is a bryophyte? Let's plunge right in. I'll take a couple of breaks during the time to answer questions. So accumulate your questions and we'll break up the presentation a bit with that. But first of all, we got to talk about what a bryophyte is. Most people have some idea and have seen them, but probably don't realize just how many there are around and how much variation there is among them. They are green plants, and in fact, they're land plants. They are related to the vascular plants very closely, not to mention they're related to the green algae along with the vascular plants. So they're green plants. They photosynthesize, they're completely plant. They're not very similar to lichens in their habitats, but they're a very different kind of biology than lichens. There's a lot of things that people might think are bryophytes, but they're not. The very first bryophyte I ever collected was a Slaginella, you can see on the left here. And I took it to my professor and I said, hey, look at this nice moss I found. And he said, well, why don't you try to key it out and see what kind of moss it is? And then I finally realized it was a Slaginella. Best way to learn. So I've never forgotten it. But so you got, these are club mosses. And as I mentioned, there are lichens, which share habitats, also quite interesting and beautiful, but are not directly, well, they're not related at all, even though they do share some really interesting ecological features that I'm going to be going over later. So our National Society for Bryophytes actually includes lichens too. Our National Society is the American Biological and Lichenological Society because of the ecological similarities that they have. As I mentioned, they're closely related to vascular plants. Together, the Bryophytes plus the vascular plants, which you all work with more, which would be the ferns and conifers and flowering plants. With the 3 Bryophyte lineages, they comprise the modern representatives of the land plants. This is one of the big lineages of life that moved on to land about 450 or 500 million years ago and diversified. It's had lots of extinction, but the 4 major lineages that are left alive that we can see around us today are the vascular plants, the hornworts, the mosses, and the liverworts. And 3 of them are placed together and called the Bryophytes. But if you look at it with this picture, what biologists like to point out is that these Bryophytes are 3/4 of what it means to be a land plant. They diverged early. And I'm going to go over the differences among the different groups. But for here, I just want to point out that we're dealing with a group that has kind of a biphasic life cycle. Maybe it's a little schizophrenic. It has a very distinct and prominent haploid generation, which is called the Gametophyte. I'll show the picture of the life cycle in a minute. And then it has a very different looking diploid phase. Diploid means 2 sets of chromosomes in each cell. That's like humans have. And Gametophyte means only 1 set of chromosomes in a cell. So that's like our gametes have. And unlike the human life cycle where we're all diploid except for just that 1 cell stage of gametes, these have very prominent, distinctive representations of both of the 2 generations. And liverworts are, phylogeny is a little tricky, but liverworts are probably the most anciently diverging group, and they comprise of even greater diversity than the other Bryophyte groups do in terms of different morphologies and different forms. And then mosses, I'll go back through the difference. You can already see some differences here, but mosses, like the liverworts, have a distinctly different and almost co-equal haploid phase and a diploid phase. Gametophyte, the way to remember it is Gametophyte means gamete bearing. So that's the plant that has the gametes. And Sporophyte is spore bearing. So that's the plant that makes the spores. I'll show you how this relates to the vascular plant life cycle in just a moment. Then we have hornworts, which are quite interesting. Maybe not quite as phonogenic, but they're coming out now. A lot of them are annual in California, and you can find these around on the banks. Just maybe they're fried due to the cessation of rain, but they were started at least in January. I haven't looked to see how they're doing. And then you have vascular plants. And of course, if you've ever grown ferns, you know ferns grow from a liverwort-like Gametophyte phase. It's called the prothallium. You see all these little blue-green bits here that look as though they were liverworts. They're really not. They're a part of the fern life cycle. They're haploid. They bear the gametes. And the brand new sporophyte, which is the leafy fern, will grow up out of mom. So there's a fundamental homology between the life cycle of vascular plants and bryophytes. There's just a change in balance that's happened. Here's a diagrammatic view of the life cycle. It's a review for some of you, but just to make sure we're all on the same page, this line divides our distinction between diploid or 2 sets of chromosomes in each cell here and haploid above here. So if we start with like a nice leafy moss plant, they bear gametangia. So they're the Gametophyte. These gametangia are of 2 kinds. There's the female gametangium, which has an egg cell that stays put. And there's a male gametangium that makes a swimming sperm cell that has to find its way over to the female gametangium and fertilize the egg. The egg stays attached to mom. So there's a form of internal fertilization and parental care going on here. Then as the fertilized egg begins to develop, it stays nestled down inside of the female gametangium and inside the mother tissue, and eventually grows up out of it. Kind of looks like an extension of it, but it's really a new generation just embedded where the gametangium was. Then meiosis happens in the sporangium to make spores, which are now haploid again. They grow out, make this thread-like structure that's called a protonema, and from that grow the leafy gametophytes. So this is the balance between the haploid and diploid generations that I mentioned, and those two generations are different from each other. So we call it a alternation of generations life cycle. And as a reminder, here's the fern one. Fern, which is a vascular plant, but still shares primitively some aspects of the Bryophyte life cycle. Here's its haploid part up here, these heart-shaped prothallia, fern gametophytes that also have the two kinds of gametangia, male and female. They have swimming sperm. They fertilize the same way the Bryophytes do. They stay attached to mom and grow into the new diploid, which is the new sporophyte. And it, unlike the Bryophytes, the diploid phase gets really big and complicated. Gets complicated leaves, many leaves, and so forth. So there's definitely a shift in balance between this one and the moss one, but it's fundamentally the same. So here is a diagrammatic view of what's going on here. At the far left, if we go back to the green algae, and this would include the green seaweeds that you see down on the coast. Most everybody knows ulva, the sea lettuce. Probably people know codium, which is called dead man's fingers. It's a really interesting green alga that grows on the rocks down there. So those green algae are the ones related to land plants, are all haploid. Their whole life is as haploid organism with one set of chromosomes. That's what one N means, one set of chromosomes per cell. The only cell that those green algae have that's diploid, 2N, two sets of chromosomes, is right after fertilization. They have a cell that's universally called a zygote. That's called the same thing in animals as well. The first thing that happens after gametes fertilize is called the zygote. In the green algae, that just undergoes meiosis right away and goes right back to haploid. So they have no diploid generation. Then we have the Bryophyte life cycle that we've just been going over that has an alternation of a prominent haploid and a prominent diploid generation, more or less equal balance between the two. Then we have the fern life cycle, which still has a free living haploid phase, which is that prothallium, but it's mostly sporophyte. Almost everything we know and love about ferns is the sporophyte generation. All those leaves, all of that stuff is diploid. Then we get to the seed plants, which includes the gymnosperms like conifers and all the flowering plants. They still have the same life cycle, but it's completely reversed basically. And everything that we see with them is diploid, just like us. Their haploid phase is buried way down inside the cone and inside the pollen grain. It's still there, but it's captured by, enslaved by, however you want to say it, by the sporophyte. So the haploid generation is completely inside the diploid generation. And it's very hard to find the haploid phase in a flowering plant. They still have them, but they're hard to find. So you've had a complete flip over evolutionary time from a 1N or haploid dominated life cycle to a 2N dominated life cycle. And the Bryophytes are kind of intermediate position in this. So here's one of these places where I can look at chat and see, does anybody have a question they would like to ask about life cycles? Does anybody have an opinion on why it might be that the big complicated organisms like vertebrates like us and the big plants are diploid, whereas the smaller organisms on Earth are mostly haploid?
If you're going to ask a question, because
Sure.
I can only see a few people at a time, you'll have to use the reactions button. and hit the raise hand, because that's the only way you'll get to the top of the screen so that I can see you, or Professor Mishler can see you.
I can see chat.
Now there's nothing in the chat, so I don't see any raised hands. So
Okay, everybody's cool with this little lecture portion on the facts of life. Okay, it is important though, because it'll come back into the story later of why it's actually important what the genetics are of their actual life cycle. Okay, so we'll go on and let's talk a little bit about the structure of these groups. How would you recognize them in the field? You certainly see them when you take a hike, basically anywhere. But how do you know whether you're looking at a moss or a liverwort in particular? So we'll first look at mosses. One hint is going to be this little cap that's on top of the sporophyte. It's called a calyptra. It's only found in mosses. The liverworts have a very different sort of a thing. The mosses are leafy here, which doesn't perfectly distinguish them from liverworts because some liverworts are leafy. But certainly if you see one of these stalks sticking out with a cap on top of it, this calyptra, that's definitely a moss. Here's what mosses look like if you look at them up close. They have leaves. The leaves are quite complex. They have anatomy. There's a lot of taxonomic characters. When we use moss keys to identify a moss, most of them will refer to the leaf characters. This one has a serrate margin. Many of them have a smooth margin. This one has a single midrib. Some of them have double midribs. Some of them have no midrib. Some have long, skinny leaf cells. Some have little square leaf cells. Some have hexagons. Some have circles. There are many, many morphological characters, and they're actually, despite their reputation of being hard to identify, they're actually quite easy to identify. There's lots and lots of characters to tell mosses apart. You just have to get, like any other new group you start with, you got to get the terminology under your belt of what the keys are asking for. But once you get that, you can, to me, it's much easier to identify a moss than it is, let's say, a sedge or some of the difficult groups of flowering plants. They have an awful lot of characters. One thing that lets you know that you're dealing with a moss, I got a better picture of it in a minute, is the opening of the capsule has these beautiful teeth that are called Peristone teeth, just unique to mosses. Only mosses have those. We've got some pictures of those coming up in a minute. There are many, many different growth forms of mosses. Some of them are bulbs on the ground, look a little bit like an onion maybe. This is not a moss here. That's actually a liverwort. This one's a Mexican wedding shirt liverwort. It has frilly pads on it, kind of like the front panel of a Mexican dress shirt. Many different looks and so forth. So it's really not that hard to actually tell these apart. This one is Politicum, which many of you know. If people know mosses, that's one of the ones they know. And it's a hair cap moss. It's got these hairs on top of that calyptra that I mentioned. Nice square capsules. So here's a diagram of the parts of the moss so you can tell them apart. The stalk that holds it up is called the seta. There's that calyptra I mentioned, which is the little pixie cap that has various shapes on it that's on top of everything. Where the spores are born is inside this part, which is called the capsule. That's where meiosis happens, and you go from diploid back to haploid. And then this dunce cap is going to come off. We didn't label it here. That's called the operculum. The cap that sits on it, when that comes off is when you see the peristome that I mentioned. Sometimes the capsules are upright, like this one. See how this is sticking straight away from the moss stem, whereas others of them are nodding. This is a key character. This is the family Bryaceae, which is very easy to recognize in many ways. In one way are the sporophytes that are nodding over here. There's the operculum, and the peristome is just inside of it. the collector is gone. We have these pear-shaped capsules. Just the shape of the capsule itself can give you a lot of the features you would need to identify them. And here's some scanning electron micrographs of what the peristome looks like. The peristome has the same relationship to classification that the flower does for angiosperms. The higher taxonomic groups of mosses are mostly defined by the peristome features that they have. We even have peristome formulae, like you would have floral formulae. We have double peristome mosses like this one here that has this outer chunky triangular row here. Then it has this inner row of peristome teeth that are very elongated and much longer. That's endostome, exostome. Some mosses only have a single row of peristome teeth. Some of them have high twisty peristome teeth. This is Centrichia, the group I work on. Its bottom part of its peristome is united together in a basketwork looking arrangement, and then the top of the peristome is free. All these little dots all over the place here are all the spores coming out for scale. The peristome moves with respect to water. Most mosses close up when it's wet and they open up when it's dry. And so the peristome is thought to regulate spore dispersal in that way, like an intelligent salt shaker that opens and closes its pores when the time is right for salt to pour out. There we go. One of the key characters that we use in the keys, one of the first key leads you're going to come to, so I'll show you that, is this distinction between acrocarpus and pleurocarpus. It's basically like tree versus shrub or something. It's a major habitat classification. The acro referring, it's what the root acro, which in Greek means on top of, like acrobat. So acro means, and carpus is fruit. So the mosses that grow in tufts and have their sporophytes born on the top of the stem, those are the acrocarps. Whereas the ones the mosses that kind of lay in mats, they lay sideways and they have their sporophytes coming off along the stem, those are pleurocarps. So we have acrocarps and pleurocarps. It's a major division. It's not a phylogenetic one, but it's a really important key division because easy to see. And there's about half the mosses are in California in one, about half are in the other. Okay, so let's talk about liverworts. Liverworts are more diverse in terms of their form than mosses are. At least all mosses are leafy, even though they vary a lot what kind of leaves they have. There's two basic flavors of liverworts. There are ones that are leafy, but there are ones that occur as a flat sheet of tissue, which is called a thallus, Thallus. You have to say that very carefully. Astarella is one of our famous Mediterranean climate California liverworts. I'm sure you guys have seen this one. These little umbrellas are where the gametangia are born up under them, but they have these white scales that surround the sporophyte that's going to dangle down from them. So when you see a liverwort with these white scales, you're pretty sure that you're dealing with an Astarella. These are quite beautiful plants. By contrast, you see this moss creeping in along their feet there. You can tell that's a moss because it's all leafy and upright. The structure that bears the Archegonia is called the Archegonia 4. So everything's haploid that we're looking at here. The sporophytes, if they're there, they're tucked up inside that white nest of protective scales. And here's a different one, conocephalum, which literally means in Greek cone head. Those of us who are old enough to remember the early Saturday Night Live, the cone heads, that's what it reminds me of. It has a very cone head shaped carpocephalum. Now, the These flat ones, the thalloid ones, are of two subflavors. Those of you who've ever cut a cross section of an angiosperm leaf would see that there are some definite similarities between what the cross section looks like of a liverwort and what an angiosperm leaf looks like. There's a bunch of tissue differentiation. You have a layer of the thallus. This is a cross section, a layer that has the chlorophyll, and it's where photosynthesis is going on. Then you have a storage layer. You have epidermis. There's 5 or 6 kinds of tissue in the thallus, which is why we call it complex thalloid. So a complex thalloid like Astarella or like conehead here, Conocephalum, this one loves to grow in gardens and things. You probably have this one growing around wet places in your yard, or you very well could. It has the most pronounced alligator looking surface, so it's kind of easy to tell that way, and it has a very distinctive smell. But the complex thalloid is called that because it's differentiated into many types of tissues. Then there is another type of thalloid liverwort, including this Mexican wedding shirt liverwort that I mentioned a bit ago, that they're not leafy, they're a flat sheet of tissue, but they have no tissue differentiation. If you make a cross section of them, the cells are all the same, top, middle, and bottom. So we call these the simple thalloids. So you have the complex thalloids, the simple thalloids, and then we have leafies. Honest to God, leafies, and most of the species are leafy. So this doesn't look much like any of the ones we were just looking at. Yet they're pretty characteristic. This one, Frulania, you may recognize. It likes to live on tree bark in the sun and has this very dark burgundy color to it, which is quite characteristic. They tend to be bilaterally symmetrical. So even though they're leafy, they're kind of easy to tell from mosses. Mosses tend to be upright, or at least have the leaves arranged all the way around their stem, where the leafy liverworts are flat and dorsoventrally symmetrical. So with a little practice, you're going to be most confused between the leafy liverworts and the mosses. But with a little bit of practice, you can figure it out. The liverworts never have that midrib. Liverworts are very often divided into 2 lobes instead of a single lobe. There's a 4th kind that is around in the spring here. You look on bare ground for this. I often find it in picnic areas that are not too heavily used. So the ground is bare, but it's not too heavily used. And these have a simple thallus, but they have their gametangium in these paper bags. There's 1 gametangium in each of these. These are the females. And then these wine-colored ones here are the males. Many mosses, many liverworts have the males and females on separate types of plants. So this scene, I get this in a lot of the Bay Area parks, and that can be around your house too. It's an extreme ephemeral. It loves the spring, and you're only going to see it in, let's say, February and March, and then it'll be gone. Quite an interesting plant. So the sporophyte, really, really different than the moss sporophyte I showed you a minute ago. It has a watery seta, its stalk, watery, not like a broomstick. This is more like just a mushy, almost like a mushy stem of a very ephemeral mushroom. And this black olive looking structure is the capsule, and they burst open into valves. So this is a picture of one of those that's burst open, and they explode the spores out of them. No peristome. They're just completely different mechanism to disperse their spores. So when you see the sporophyte, you absolutely know that it's a liverwort. We had some hints this was a liverwort because how flat it is and seeing the leaves coming out on each side of the stem. But when we see the sporophyte, we're like convinced. And then check this out. I borrowed this picture from David Wagner recently, who takes beautiful photographs by using a microscope that is able to move a little bit and photograph, and move a little bit, photograph, move a little bit more. and then informatically stitch the pictures together. It's called a stacked photograph. It gives you great depth of field. This is what it looks like inside of the liverwort capsule. There's two things in there. There's the spores, and the spores have fantastic ornamentations on them that are quite useful in classification and identification purposes. And then they have these spring-like cells that are called elaters, E-L-A-T-E-R-S. Those are really are springs, and they're under pressure inside the capsule. So if I can go back here, what blasts open this black olive into this black cross here is the pressure of all those elaters want to spring open, like you have a spring held tightly closed. They want to spring open. And when the capsule dries out, they do. You can see the elaters still hanging on here. It looks a little bit fuzzy, like it was felty. Those are the elaters have stayed in place, and the spores have been blasted out into not outer space, but at least inner space. So completely different than mosses. Mosses don't have elaters. Mosses have a peristome to regulate their spore dispersal. They don't blast open like the liverworts do. So they're very, very different. Okay, a little bit of ecology. Where do they grow? We do think that they grow mostly or maybe only in moist shaded places. They do like that, but if you think that they're only found there, you would be wrong, because they occur pretty much everywhere. Every terrestrial biome. They don't live in the ocean, but they're in alpine, they're in forests, they're in all kinds of deserts. The mosses I work on are desert mosses, shrublands, alkali seeps. Pretty much any terrestrial biome will have bryophytes in them. And the ones that live in strange places are almost the most interesting ones. They can grow right in the water. So we have aquatic species. They like rocks, trees, dirt. Many of them just grow in soil. The ones that grow on rocks and trees tend to be specialists, where they only grow on certain kind of trees, or they only grow on limestone rocks, or they only grow on acid rocks or something like that. And one other thing that I'd like to point out to people when I take CNPS groups out for hikes is when you stand at any one place, you often will be in sight of more bryophytes than there are vascular plants. In any given location, especially in our East Bay Hills, there's a lot of bryophytes. So in a forest like this, which would be something you could see in Marin, you know, you have a certain number of vascular plants there, but each of the vascular plants has a whole community of different bryophytes on it. And this big shaggy pleurocarpus moss here, Dendroalcea, looks like a little fir tree. That's the species that, if we do pursue California getting a state moss, that's probably the one we would go for, because most people know that one. But here's one that loves to grow on rocks in our area. Grima levigata is on boulders right out in a grassy, disturbed field. It'll be there. Fontanalis is one of these ones that grows underwater. It's called the fountain moss. It only grows in really clean water. Rixia is a strange complex thalloid liverwort that likes to grow in various kinds of habitats, but most of them are dry, quite dry.
Brent, I accidentally muted you.
That's okay. I was going to stop right now and just see if there's any questions, because I'm about ready to go into another portion of the talk. And I wanted to see if there was any reactions to what we talked about so far.
There are a couple of questions and comments in the chat. Paul Barnett is suggesting that perhaps diploid have more redundant genetic information for adaptation.
Yeah, you know, that it's speculation, but that's what the experts actually speculate. The idea is the one advantage that diploidy has over haploidy is that you can have dominance and recessiveness. So as everybody knows, we humans are affected if we have close relatives mate with each other, we can end up with rare deleterious alleles actually coming together and being homozygous, which can cause defective offspring, maybe not even viable offspring, or at least offspring that have genetic problems. And that's because a diploid can have one bad copy, and probably each of us has some defective alleles in our genome that don't work, but we're saved because we have at the other position in the other chromosome, we have a functional gene that makes what we need to live. And when you get these homozygous detrimental alleles when close relatives mate, that's when you have two dysfunctional copies that are in the same organism, which is often very bad medically. So only diploid organisms can do that, can have dominance and recessiveness. So Paul's right that that's thought to be why each of the lineages in the fungi, in the animals, and in the plants, each of the lineage that got big and complex also transformed to being diploid, all from a haploid starting point for everything. Most of the world is haploid. I think there's 7 or 8 different times evolutionarily that diploides evolved, and every time has resulted in big complex groups like the metazoa, the group that we belong to, that includes us through insects. Bruce pointed out the CETA looks vascular. It's actually a bunch of cells. In the moss sporophyte, there really is vascular tissue. We actually think that the mosses share vascular tissue with the vascular plants. So they're even in a homologous sense. This is a little bit of a controversial position, but there is xylem and phloem or else tissue that looks exactly like xylem and phloem inside the CETA of the moss, but not of the liverwort. Liverworts look like they don't have it, never had it. So that's kind of interesting. Yeah, there are, as Paul points out, there are species with high numbers of chromosomes. And some of our most successful weeds are polyploids. So polyploid is the name for the case where the genome is duplicated. And sometimes after you have hybridization, if you duplicate the genome, there's a hybrid vigor, and a lot of our weeds, which you have to say weeds are pretty darn fit. They're too fit in some ways, but their polyploid is associated with diversification. Yep. So it might not be the polyploidy per se that's adaptive, but by having more copies of things, more copies of your genes, you have room to wander, if you know what I mean. If you have 2 copies of a gene, you can experiment with one of them. If you have 4 copies of the gene, you can presumably explore with 3 of them. And then occasionally it hits on a mutation which is random, hits on something which actually is better functional. So every once in a while, you get a novel trait that is a good thing. So that's how evolution happens pretty much. Yeah, Vernon asked if I'm going to say anything about the sphagnales. What I wasn't planning to, but I will. The group that most people would recognize would be sphagnum. Sphagnum is a moss. In an evolutionary sense, sphagnum is the sister group of the rest of the mosses. So the deepest evolutionary divide in the mosses is between sphagnum and everything else, Politicum, acrocarps, pleurocarps. So they've been separated a long time, hundreds of millions of years probably. And sphagnum has its own set of very unusual but great adaptive features that are actually quite interesting and they have, you could say, analogs in other mosses, but sphagnum has really gone in for a particular lifestyle and is actually one of the dominant groups of plants in the world. When you get to the northern parts of the planet, sphagnum is definitely an ecosystem dominant plant. Okay, so we'll go back and I'll share the screen again. What I wanted to do was, let's see if I can. What I wanted to do was give the mosses are from Mars, vascular plants are from Venus part of this, where I compare the biology of bryophytes with the biology of trachyphytes, which are vascular plants. And we've been doing that a little bit already. We've been talking about this first item in the list, the haploid dominance versus diploid dominance. We've already been talking about that quite a lot, but I want to go into some of these other features. And the take-home message from all of this is that the bryophyte groups are perfectly well adapted for what they do, but they're just following a different whole strategy from the vascular plants. So they're being a land plant in a very different way. So I'm just going to run through these in order. The haploid dominance, we don't need to talk about too much because that's what we've been talking about all along. Without those genetic benefits of dominance, presumably genes that are in the gametophyte are more obvious to selection. So if there is a deleterious allele present in the plant body, it can't be hidden by dominance anyway. So there may be severe selection going on to kind of get rid of some of that variation. As possibly a counterpoint to that, if there are mechanisms that are tending to cull out a lot of the genetic variation that might stay around if it was a diploid, it turns out that the bryophytes have just this incredible amount of plasticity. All plants do this. Vascular plants certainly do it, but the bryophytes do it to a major extreme where the morphology that they show can be quite variable, which is one of the frustrating things about taxonomy sometimes until you get a good handle on that. So there may be some interaction between the amount of genetic variation that you have and the amount of phenotype variation you have. You can still have lots of phenotype variation as long as you have a lot of plasticity. If one genotype can make many different morphologies and have different physiologies in different environments. The most important area in which they differ, and the word for tonight, is poikilohydry. My students think I'm crazy. I use this word a lot. So poikilo in Greek means changeable. When you probably remember in general bio, when you talked about a reptile, the scientific term for being cold-blooded is poikilothermic. So snakes and lizards are poikilothermic. And what that means is their body temperature changes with respect to the environment. So if the lizard's in a cold place, it gets cold. And if it's in a hot place, it gets hot. It doesn't have the regulatory mechanisms that a mammal does or a bird does. So poikilohydry is the same kind of thing as poikilothermy, except it's referring to water. So a poikilohydric plant rapidly equilibrates its internal water content to that of the environment. It doesn't buffer things to the extent that vascular plants do. Vascular plants have a cuticle on the leaf surface, maybe punctured by stomates, but the stomates can open and close to regulate the movement of water. They have roots and vascular tissue that are bringing water up inside the plant. So you can have quite high water content inside of a plant, even though the outside environment is really dry, really, really dry. It'll take a long time for the plant to feel that at the tissue level until it runs out of water down at the roots. It won't dry out just because it's dry in the air. Whereas poikilohydric plants, and all the bryophytes are this way, also the lichens, this is one area where lichens and bryophytes share, they don't have roots and they're just bringing the water in and out of the plant surface directly. And it's also hauling a lot of stuff in with it. So I don't know if I have a different slide for it, but poikilohydric condition is going to mean that you're going to be quite sensitive to pollutants, and the bryophytes are. They're sensitive to air and water pollution because they bring particles right inside the cell directly from the outside as water is coming in. So they're really, it's bad for them, but they're really good environmental indicators for us. The water quality and that sort of thing is easily monitored by what bryophytes can live in a place. And then one of the things I study myself, which is related to poikilohydry, but not the same, is desiccation tolerance. Desiccation tolerance is a different thing than drought tolerance. Drought tolerance is being able to tolerate low levels of water in your tissues, and many vascular plants can do that. But desiccation tolerance is being able to come back from zero water in your tissues, and very few vascular plants can do that. None in California. There's a few handful of vascular plants that can do it. Well, I should say of angiosperms. There are some ferns in California that are quite desiccation tolerant. And then there's our good old club mosses that are desiccation tolerant. But the angiosperms at least, and most of the ferns are not desiccation tolerant. So here's a beautiful moss, which has mostly tropical relatives, but it does occur in our flora. Hookeria has these, obviously, this only occurs in really moist, wet, dripping forests like redwood forests. Beautiful cells, and just illustrating how the water is able to just zip right in and out of the plant. Here's my favorite group, which is Tortula or Centrichia. This is one clump of moss that I separated into two halves, and I poured water from a canteen on the left half, and I left the other half alone. And it goes from what you see on the right here to what you see on the left here in just a matter of a minute. Some of you have seen me make this demo at field trips. If you're taking a group of kids out on a field trip, they just love to see, you can see the moss come back to life. And I think right here is actually where, if all goes well, I'm going to show you a video that KQED made. And let's see if this works. And you should be able to hear the sound from it.
Some living things, when faced with a drought, do more than just survive. When water disappears, they can cheat death itself through a kind of resurrection. Look closely at this tree, baked by the California sun. It's covered in moss, and living in that moss is a microscopic animal called a rotifer. When there's no rain, the moss and the rotifers dry out. The rotifers stop eating, the mosses stop photosynthesizing. They're reduced to a pile of chemicals, and they can stay that way for years, even 100 years. When the rain comes, the mosses spring back to life in seconds, using a rare ability. Right before they dried out, they wrote themselves a set of genetic instructions, so that if they ever got water again, they could start producing proteins and growing right away. Scientists call them resurrection plants, and they hope to use their genes to engineer crops that could repair themselves after a dry spell. So why do mosses work this way? Well, here's the inside of a blade of grass. See those tiny tubes around the perimeter? Those are for moving water up from its roots. But mosses don't have roots. Their porous cells absorb water like a sponge whenever it's available. For mosses and rotifers, it's feast or famine. When it rains, the water activates the rotifer's metabolism, and they need to fuel up. They use the crown of cilia on their head to create a current, to suck in all the algae and bacteria they can. They grind this food up with their jaws. See the jaws? They look like a tiny beating heart. Rotifers and mosses, they're experts at living without water. And maybe in this miniature resurrection kingdom, researchers will find clues for how the crops we humans depend on might survive a dry Californian future.
Okay, isn't that fun? Let me see if I can get back to my PowerPoint. We helped to make that video, but they have great skills for photography and So basically what you saw there was a couple of examples of the moss being able to dry up completely and then come back to life. Those wetting up scenes that they showed were not sped up. Those were not, fast time. They were real time. So it's really amazing. So let's look at the evolutionary distribution of that kind of desiccation tolerance. And we see it in the Bryophyte groups here. So here's the land plants, and these are the 4 major lineages we talked about earlier. Here's the vascular plants, and then here's mosses, hornworts, and liverworts. The Selaginellas, some of them are desiccation tolerant, but not all. Some ferns, but not all, are desiccation tolerant, and then no gymnosperms are. And there's a handful of angiosperms, just none that I know of in the North American flora, that are truly desiccation tolerant. The way the mechanism works, our lab works on this mechanism. To make a long story short, the moss stays ready to dry up all the time. It has stored within its tissues what it's going to need when it rehydrates. So it's able to dry really quickly without much warning, and then rehydrate when water gets added. That's why they're saying in that film, it's something like writing itself a message in a bottle before it dries out. Then once it gets water, then it bursts open all these resources and uses them to recover itself. So it really has the same kind of strategy that the experts recommend us to have for our earthquake survival strategy. You can never tell when an earthquake is coming, so have your go bag and all the things you need ready in your closet, so that if it ever gets an earthquake, you'll be able to take care of yourself. That's sort of the strategy that they're using. They don't have time to prepare to get dry, and they don't have time to prepare to fix themselves. They just have to kind of be ready to do that. So it's a repair-based strategy that's actually quite interesting. So one other aspect of water that's important, that's different than the physiology we were just talking about, is just reproducing. I showed you that the Bryophytes have a swimming sperm, just as the ferns do. They need to actually have water in the environment to swim. It's not a great system for a land plant to have. The pollen approach that the seed plants take, a lot more sensible. Swimming gamete is evolutionary holdover, you might say. It happened with the animals too. The primitive form of reproducing in our group was swimming sperm in water. And then internal fertilization arose as a way to not have your gametes out there in the environment trying to swim somewhere. So it's not a great thing, and it's kind of maladaptive, and they have trouble making the sporophyte, particularly in the dry land situations. We have many species of moss that almost never produce sporophytes, just probably because they can never get the gametes together. So we get lots of inbreeding in the ones that monoicous means that they have the male and female gametophyte on the same plant. And the dioicous means they have the male and female on different plants, like that Rixia, or like the spherocarpus we looked at. And the lack of sporophytes just mean they can't swim. Those sperm would have trouble going even an inch. So that's a very distinctive feature about bryophyte biology. Something we're discovering more and more is that the moss clump itself is the actual organism, not the individual moss stem. They're actually related to each other. So the moss clump is a little bit more like a beehive than it is like an individual beetle, given the combination of the growth and their external water conduction, which they do instead of internal water conduction. They have very efficient systems for moving water outside the plant, that the clump itself kind of acts as the individual in the environment. There's communication that goes on. We know that the moss plants share signals across space, which is what pheromones are. Animals do that too. So if one individual communicates with another individual kind of outside with a chemical, with a a hormone, we call that a pheromone. So pheromone is sort of external communication. And all of these things, the mosses in particular, are doing. Since they have a hard time doing sex because of the water limitation, they have very well developed means of asexual propagation. Marcantia, one of the ones you could see in your planters or in your greenhouses with these gemmy cups. All the little structures bouncing out of there are asexual propagules, not spores. Mosses have all kinds of structures on them that fall off and grow into a new moss. This is one you might have seen. This one's in redwood forest. Alicomnium androgenum looks like it has a sporophyte sticking up there, but it's a pom-pom, kind of like a cheerleader's pom-pom. And that's a bunch of gemmy that each can fall off and grow into a new moss plant. So the stem has been extended on into this pom-pom of gemmy. There are some mosses like Orthodicranium, which is also in your vicinity, likes to grow on trees. You can see the person's finger there. You just touch it and your finger is full of leaf tips. All the leaf tips break off of this, and every one of them can grow into a new plant. Oops. Here's a liverwort that has these gemmy clusters. These are two of my guys, two different centricheas that have two different kinds of gemmy, but they grow together on the same trees. These grow both in California. These have little, Centrichia papulosa has little round gemmy on its leaves, and Centrichia pagorum has leaf-like gemmy in the axils of the upper stem. These two guys, this is taken in a natural environment, and these two grow right next to each other. They're easy to tell apart. You'll find these on trees in the city. For some reason, they like maybe some moderate amount of pollution because they seem to really like park trees. There's one of the pagorums up there. You see it's kind of gemmy. So basically any part of the moss, the way we propagate them, I learned from a long time ago from a bonsai gardener artist. They use mosses in their creations and they just blend them right up and put them where they want them in an arrangement to get this nice arrangement of mosses in a display. They're small, they don't have roots, so they're only influenced by their immediate microenvironment, the few millimeters that are just around them. And it appears that over geological time, they're certainly not as influenced as big plants are by climate change. We have plants that linger for a lot longer. So you have bryophytes that linger in an area that are from a past era. They might be tropical from when an area was tropical, and they still manage to hang on because they have their little microenvironment. So we have a lot of interesting range disjunctions where we have bryophytes in North America most of whose relatives are down in the Caribbean, and they're probably just hanging on there for the long haul. All of this kind of adds up. The fact that there's a lot of empty space, a lot of empty habitat that they could live on, that they don't live on because it's hard for them to get established. Their vigility means how far you can travel, and establishment means getting actually established in a place once you travel to it. Both of those are pretty poor in bryophytes. Most available substrates, therefore, are not filled in. So we think that most of the selection pressures that are happening on bryophytes have to do with the physical environment, the temperature and water, that sort of thing, and not so much the biological part of the environment, which would be competition. They often tend to cooperate. One bryophyte will be able to grow better if another one is present in the area. So the microhabitats, the possible lack of genetic variation in at least selected genes, the fossil record for possibly for those reasons indicates really slow morphological evolution. You have to go all the way back to the Paleozoic, so way before the dinosaurs, to find any bryophyte that doesn't look like the modern ones. They're the things that we call genera were well established, and things that we call species were well established even when dinosaurs were around. So they're quite static, at least in their morphology, possibly not in their physiology. So that the picture we get is a group that is successful and is quite a bit older than the flowering plants for sure, but has sort of aspects of its biology that make it evolve kind of slow and have biogeographic patterns that are like those of angiosperms, but they're often at a lower taxonomic level. We have many species level taxa that are on multiple continents. So this last part of the talk, what I was trying to emphasize are the ways in which bryophytes differ in their biology and ecology and evolution from the vascular plants. And the main take-home point is they differ in just about every way possible, from genetics through physiology to morphology. So they didn't evolve on different planets. They're really not from Mars and Venus respectively, but they you could almost believe they were, given how differently they have solved this problem of living on land. And we find these contrasts that I've been giving you here are very useful with students to show them how different organisms can kind of skin the cat a different way. Different organisms can live with equally good but quite different strategies. So this is the motto: bryophytes are not just miniature tracheophytes, they're actually quite distinct. And they're definitely not primitive or lower. If you ever want to start a bar fight, go into a bar with a bunch of bryologists and tell them they work on primitive plants. Them's fighting words. The argument is that bryophytes are a very successful group of plants. There's tens of thousands of species of them. They're not less adapted than vascular plants are. They're just different. They've come up with a different set of solutions. So their natural history has some advantages and some disadvantages, just like any other group of organisms. And they're really beautiful. Here's a couple of nice pictures from Martin Hutton to finish up with. Here's our conocephalum again, the cone head liverwort. Here's one of those nodding capsule mosses, and here's a hornwort, which we didn't talk too much about because they're not as variable as the other groups, but they are quite interesting in their own way. So I just want to finish up with how do you get more information about bryophytes? The easiest way to do it quickly is to go to our CNPS Bryophyte chapter webpage, where we keep a set of resources, because we realize a lot of people are not familiar with them. So there are resources there on a lot of recaps of what I was talking about tonight, and then tools, including physical tools. How do you study them? Literature, how do you collect them? How do you make a specimen? And so forth. We try to make this website a service to people who are interested in bryophytes. So there's a great source. Oh, and we have our chapter newsletter, which is quarterly, is called Bryolog. Pretty clever, huh? And Bryolog is posted up there. We don't do paper copies. We just do an electronic copy of our newsletter quarterly. There's a big project that I'm involved in that on Centrichia, which I've been talking about a little bit. That's the star moss. And you can go to that website and get a lot of information about desiccation tolerance. And the nickname of the grant is 3D Moss. So if you look at 3D Moss on Google, you'll certainly find it. And that is the group that's working on the physiological mechanisms and ecology of how this desiccation tolerance affects the mosses that live in deserts. Then there's a resource that our chapter has adopted and we're trying to work on, even though it's all volunteers. This is like the Jepson eFlora maintained in my herbarium at Berkeley. It's not quite as slick as the Jepson eFlora because we have no paid staff working on it, but we are interested. You can still key out mosses here. It has keys to all the mosses and illustrations of many. So check this out, and if you feel moved to help out in it, we're always looking for volunteers. But you can find that on the University and Jepson Herbaria webpage, under e-floras, you have the Jepson e-flora and the moss e-flora. Okay. So we have one final time for questions, or I can look at the chat to start with. Yes, you could find mania under. They really like it under chaparral. I think the reason they like it is they like the bare zones that the animals keep clear around the base of chaparral shrubs. So that's actually a pretty good place for liverworts. That's a good place to go look for these annual liverworts like Rixia and Spirocarpus and Fossembronia, the Mexican wedding shirt liverwort. So yeah, chaparral is actually pretty good in the spring. Woody asked, if they don't have roots, how do they affix themselves to the substrate? They have rhizoids, which are analogous to root hairs. So they don't have roots, but they have little unicellular extensions that come out of their surface, which both attach them to their substrate and also function in bringing in nutrients, not water, but nutrients. So they have rhizoids. They're not homologous to root hairs, but they're quite similar to root hairs. Ian S., if bryophytes have any known medicinal or toxic properties? A few. There's not a ton of work on that. One of my graduate students was interested in this topic and worked on Chinese medicine anyway. And he actually went to China and did field work. There are quite a few bryophytes that are used in Chinese medicine. And you can find them even in San Francisco in Chinese herbal medicine shops. They're topical, many of them. And the few toxic ones that I know of are like skin irritant kinds of things. Some of the sphagnums will do that if you stick your, I know workers that are working up in the North Woods, if you get sphagnum on your hands all day long, it might irritate them. Sphagnum is big business. Somebody was Vernon was asking about them. Sphagnum are by far the most economically important of the bryophytes. Not only are they collected to be used in agriculture, so we can buy sphagnum moss at our local nurseries, but they're also used for medical products. They're used for bandages, feminine hygiene products, anything that you want to be sterile and absorptive. So the big pharmaceutical companies have mining operations. They're pretty horrible. They're like strip mining, where they just take the whole surface of a bog and they sort out the sphagnum, and then they sell it to Menon or some other company. And apparently the different species of sphagnum are useful for different things. They are quite antiseptic. Native cultures have been using sphagnum for years as a diaper or a bandage or something like that. So there are maybe not as many as angiosperms, because I don't think they're chemically quite as complex, but there are quite a few medicinal and cultural uses of them. Janet asks, in the moss flora, what proportion are cosmopolitan? You know, part of the problem is our taxonomy is a little bit messed up still. The many species in California have the same name as a species in Europe or in the Eastern US. But as they're being looked at with molecular methods, sequencing the DNA, it's often found that what is called the same thing is really a relative of that other plant, but it is quite a distinct thing. So we actually have a lot more endemic lineages to the west than you would get at by looking at the taxonomy. But even so, there is a sense in which the average bryophyte species has a bigger range than the average angiosperm species. We do have some cosmopolitan ones. We do have some very rare ones, but the most narrow endemics in California, there's a few that are really, really endemic to just one particular hill, but not nearly as many as there are angiosperms. And there are even very few that are restricted only to California. So let's say 6 or 8 of the California flora are these highly restricted endemics. And then we've got a bunch that are Californian and maybe not outside, then a lot of West Coast taxa. Our flora right now, I think, is about 800 and something species. But that's, like I say, that'll increase if we actually were able to dissect them genetically. Yes, Hillary, peat moss is the sphagnum. It's another name for it. Oh, serpentine. Yeah, Kevin asks, interestingly, I worked on that myself one time, and the serpentine loving species seem to be the same as the calciphiles, which is a very strange thing because there's not a lot of calcium in serpentine, but it might be something physiological about the effect on them that they have. So that it would be interesting to look more carefully at the serpentine species, because some of them also live off serpentine, but they may not be genetically the same lineages. And then Clinton asked how many species of sphagnum are growing together in the same bog. In the West, not many. We don't have that many sphagnums in California. There's usually just one. The Sierra has lakes with sphagnum, but usually just 1 or 2. When you get up into the north, and then when you get into the northeast, let's say upstate New York, there can literally be 35 species of sphagnum within a couple of feet of where you're standing. It's quite diverse in bogs up there. For some reason in the West, not so diverse.
Brent, I have a question about desiccation. I'm curious how they stay alive for these long periods. Do they have a way of regulating their metabolism, or?
Well, they're completely inert. So I was the one that gave that the KQED people that bag of chemicals idea. So they're about as inert as a living thing can be. It depends on how you define life. The people that define life as equaling metabolism, you would then have to say that they're not alive, that they literally are coming back to life. There's no mechanism to, whatever mechanism they had to protect themselves. One of my graduate students works on these sunscreen chemicals that they have that do protect them while they're dry. They tend to get very black in color or have prominent, they're flavonoids, some of the same chemicals that angiosperms use for sunscreen, because too much radiation is bad for a living thing, as we can find out if we don't wear our sunscreen. And even plants can suffer, even vascular plants can suffer from having too much radiation. So it's a common problem that plants have. But whatever the moss is going to do, it's got to do it before it dries, because it has no mechanisms operating while it is dry. So part of whatever chance it gets to protect itself before it dries completely, it's putting on some sunscreen basically. And then it just sits there. It has to just sit there and take it. And eventually it will die, because chemical bonds will over time break, which is why inert chemicals that you have sitting in your medicine cabinet will eventually expire, even though you never open them. They just expire because, chemical bonds break, and pretty soon your antibiotic's not working anymore. And so that's the remarkable thing. They actually can prepare themselves a little bit, but they don't get much warning. They only get a 30 minutes or something warning that they're going to dry. And then they can be dry for, the record is based on a herbarium specimen that was revived, was 100 years. That's herbarium. And there was a recent story up from Canada, a group of people working, the glaciers are retreating so fast in Canada that the Canadian botanists are studying what's underneath them. So new land is coming out from under a glacier that's been covered for at least thousands of years. And darn if they didn't find that the Bryophytes that had been covered by the glacier and essentially, dead, they look like litter, would regenerate and grow. So who knows? I think they're recovering some angiosperm seeds too. So that, I mean, it's global warming is not a bad thing, not a good thing, but it is giving researchers a chance to see what was under the glaciers of the Pleistocene. So it is one of the most remarkable things about it. They can dry up completely and they can come back. It takes them 15 minutes to get back to normal. So it is amazing.
Another question would be, is there any horticultural potential in bryophytes?
A lot of people, it's ornamental. They don't have a lot of caloric value, so they're not going to be good for food. They're certainly good for roughage. If you need some roughage in your diet, you can eat some, but they're not going to be much nutritional value. They're also not poisonous. But where the horticulture comes in some countries, even in Eastern US, and especially in Asian countries, especially in Japan and Korea, the horticulture is fantastic. They're used to make lawns with. People don't have lawns of grass. You make beautiful moss gardens in the right environment. People in the Carolinas do that. There's quite a movement afoot now to tear out your grass and have a moss garden instead, because you don't have to mow it and you don't have to water it. It can just dry out and come back to life. So it'll be ornamental horticulture where the applications are, I think.
Is there actually a source for various types of mosses, or?
There is. Yeah, a friend of mine actually has a company, Mountain Moss, I think it's called. You can buy it, but she works back east, so it's not really for our environments. I don't know of a company that's selling it, and you would have to have it ethically produced. You know, you don't want people going in and stripping the forest, which sadly the Forest Service does let people do. It's called forest products. And so if you can prove you're not a big corporation, you're kind of a local person, you can get a permit to actually go and strip mosses and lichens off of trees, which is horrible. So what Annie, Moss and Annie, who's the lady I'm talking about who owns this business, they do it rescue style. So what they do is they go to some place which is going to be developed or cleared off, and they rescue all the bryophytes off of it, and then they grow them. And if you're doing something like that, then what you want to do, if you want to grow them in your yard, you want to find a habitat match. It's just like trying to pick out the right place to grow a vascular plant. You want to match its natural environment if you can. So if it's a shade-loving species, put it in the shade. If it's on limestoney kind of soil, then you're going to have to probably amend your soil. But it really works pretty well. The blender idea works fine. You can make your own, you can buy bags of moss seed. And what moss seed is just a bunch of moss that's been ground up. And then you put it out someplace and it'll grow.
Kevin has a question in the chat.
Yes, there are some. The really poster child for that in California is Funeria Hygrometrica, Funaria. Funeria loves fire. They were doing controlled burns up in Yosemite National Park in the Wawona side. And when I went back the year later, the ground is just literally covered with this one moss. I'll write it in the chat if you want to look that up. That it's partly in a spore bank, probably. There are some spores that are down at like a seed bank, and then it disperses in. But for whatever reason, it just loves after a burn. When you find it in an intact forest, you look around and almost always it'll be at the site of an old campfire in that forest. And there's others too. As Rebecca points out, there's only a few mosses that will grow well in all environments. Funeria is actually one of them. In general, you want to pick ones that are growing in nature in the kind of environment that you want them to grow in. There's one moss, the weediest moss in California is called Ceratodon. There's how you spell Funeria. Ceratodon is, I call it the freeway moss. It's the one that's just literally growing in sidewalks, and you find it in the cracks of off to the side of the freeway, or that'll grow anywhere. If you ground that up and put it, it'll grow on really crummy, bare ground. It would even cover probably an old asphalt area you're trying to, make natural again.
I don't see any additional questions.
Okay, well, good. So I hope I taught you a little something about Bryophytes. And if you want to know more, Bryophyte chapter does a lot of activities. We run a lot of walks and field trips and classes. So you're welcome. Anybody's welcome to join any of the activities they would like to.
Well, Brent, I want to thank you so much. This was a fascinating presentation, and expanding our ideas of what the native plant universe is, and that we should.
Yeah, we're trying to do that. That was, I hope you guys are happy you admitted us as a chapter. We're trying to be good citizens by expanding everybody's awareness of what native plants are. And, a lot of people are finding it's a really good off-season botanical hobby, because, the angiosperms are best in the spring, whereas the Bryophytes are best in kind of the dead of winter, the rainy season. So a lot of people that mostly love vascular plants, they like to do Bryophytes as kind of their off-season sport.
I think we'll maybe get our field trip people involved in some January walks. Again, thank you. I
Sure.
want to just give you a preview of next month's presentation. It's going to be on California prairies, an increasingly rare habitat in California. And the presenter is Glenn Holstein. So look for a notification and a Zoom link for that one. So again, very much appreciate Professor Mishler, your presentation, and look forward to seeing you all next month.
Okay, thanks for having me.
You.